last presentation will be Austin from the Meyer Lab.
Hello everyone, my name is Austin Shirk, and this summer I had the opportunity of studying specific RNA sequences as potential drug targets in Streptococcus pneumoniae. And I conducted this research in the Meyer Lab. Now the significance of this research is that in a medical setting, Streptococcus pneumoniae is known to have increased antibiotic resistance, and so additionally over 400,000 hospitalizations per year in the U.S. Is attributed to strep infections such as pneumonia or ear infections, etc. So to give a quick outline, first I will be giving a brief background of our potential drug target, which is called a riboswitch. Then I will discuss the significance of studying the riboswitch structure and mechanism. Next, I will be going over my methods for identifying the riboswitch signal. And lastly, my findings. So when we're looking to find potential drug targets, we want to specifically target the pathogenic microorganism and not the host, which sounds obvious, but it demands clarification. And so there are various ways that antibiotics can target an organism, and there are various molecules that are locations for drug targeting, including including DNA, RNA, and proteins. In the Meyer Lab, we are specifically studying RNA structures as drug targets. So the specific RNA sequence that I'm studying is known as a riboswitch. And riboswitches are gene regulators. And you can see the nice diagram shape of a riboswitch, super simplified. It's located at the 5' UTR region of an mRNA strand. And so I mentioned it's a gene regulator. So downstream of where the riboswitch is where the gene is expressed that's regulating. And so this comes in handy when riboswitches control essential control genes that are encoded for essential proteins for cellular growth. So when thinking about drug targeting, it would be ideal to target the riboswitch to prevent downstream expression of essential proteins for cellular growth. That way we can diminish cellular growth in pathogenic microorganisms that are infecting our host. So there are many characteristics of riboswitches that have yet to be discovered, but are essential to learn further to develop drug target plans. But we do know that riboswitches receive signals from what's called a ligand. So for example, when the ligand binds to the riboswitch, downstream expression is terminated. So the gene downstream is not expressed. Again, having that drug target mentality, we would love in the future to have a drug mimic the ligand so that when the drug is targeted to the riboswitch, it binds to the riboswitch and prevents downstream expression, hopefully of genes that encode for proteins necessary for cellular growth. So my project this summer was to identify the ligand that binds to our riboswitch at location SP1384. A little bit about the riboswitch that I'm studying. We know that the ligand, we're expecting it to be an amino acid, and we also believe that the riboswitch that the gene that is regulated by our riboswitch is an essential gene. So this search for the ligand will give us insight as to how to best target our riboswitch. So I will be conducting, I conducted Miller assays which assess gene expression levels. So how does that relate to my riboswitch? Well, Miller assays, again, they assess gene expression levels. So I'm going to be assessing the enzyme activity that's actually being regulated by our riboswitch. So in lab, we constructed a riboswitch fused to an enzyme gene, and that enzyme gene is known as beta-galactosidase, beta-gal for short. So first, with the Miller assay method, using cells that have this gene contained in them, I grew the cells on 3 media conditions, and they vary in nutrient richness. So we have poor, moderate, and rich. Next, I added a chemical known as ONPG, and what ONPG does is when it enters the cell, it reacts with the beta-galactosidase enzyme that was produced from riboswitch regulation, and this reaction produces yellow color in our samples. And depending on how yellow our samples are, is dependent on the amount of beta-gal produced. So to sum up, I'm quantifying the total beta-gal activity, which is proportional to the beta-gal amount in our cell samples. Also to note, I will be testing a mutant riboswitch, which is ligand insensitive, as a means to distinguish if any beta-gal change is specific to our wild type, which is the normal riboswitch. Okay, results time. So in the x-axis, we see the 3 media conditions, poor, moderate, and rich. And in orange, we have our wild type riboswitch, and in the gray, we have our mutant 1 riboswitch. Now what I can do with the results as seen to the right is I can compare the wild type to mutant 1 ratios of our beta-gal activity across the 3 media conditions. And so, important to note, the poor and rich media have the same wild type to mutant 1 ratio. And when we're looking at the beta-gal and moderate media, it is a 1 to 2 beta-gal ratio. Now, this could suggest that the ligand, which is an amino acid, could be in ideal concentrations in the moderate media that show a significant beta-gal change. So I further utilized moderate media, and I grew the cells on moderate media with specified amino acid knockouts because, again, I'm looking for a ligand that is an amino acid. So if specific amino acids are taken out of the media, we would expect to see a beta-gal change if it is the ligand, if the amino acid is indeed our riboswitch ligand. Okay, nice. They came up at the same time. So the first graph, we see 4 separate media conditions. The first is the moderate, which is what we saw in the last graph. And then we see the specified amino acid knockouts in the moderate media. And so when I conducted these 2 replicates of our Miller assay, I was looking at the minus tryptophan activity, minus tryptophan media, and it looks like there's a decrease in beta-gal activity of our riboswitch. So then I repeated these conditions with 2 additional biological replicates, and I did find that, again, looking at the wild type minus tryptophan conditions, it averaged out to the beta-gal seen in our moderate media. And to some, that does not conclude any ligand identification. And also important to note, I tested poor and moderate media with all the amino acid knockouts or increased amino acid conditions. So all 20 amino acids I tested. And no significant change in beta-gal activity was seen. So to conclude, my Miller assays did not clearly reveal an amino acid ligand candidate for SP1384. It is possible that my amino acid additions to the media were not taken up intracellularly. So alternatively, future plans could be with background knowledge that there is sequence evidence that our riboswitch ligand is alanine. So we could reconstruct our tiger 4 strain to have a deficiency in alanine synthesis and conduct the Miller assays again. I would love to thank NSF, Boston College REU, and Boston College for their generosity and the amazing opportunities. And I could not thank enough my PI and my Meyer Lab team. So. Yeah, so that's a great question. So there are a lot of different types of riboswitches. And so I'm specifically studying what's called a T-box riboswitch. And a characteristic of T-box riboswitches is the ligand is, at least from what I know, an amino acid. And so that's why we were specifically modifying the amino acid. Thanks.
Okay, so we'll have a break till 4.
And the snacks are down here. Where the pizza was. Why? Right. Come, let me see if I got it right.
Good job, Austin.
Push through. Do you know what's different about
Good, yay, happy for myself. Where are we going?
I'm gonna make sure I got them all out. That's Let's check it. This is not you. That's you. And that's the most recent one. So all you'll have to do is click that. And you'll be ready to go. And I'll try to get it ready. And then the other, let's check it. So I have Courtney. I'll be ready to go. We won't even have to switch computers. Get some water and breathe. Excellent job. Everybody's doing so well. It's wonderful.
It makes it really nice when all the presentations are on the computer.
Right? And so that you don't have to, I can't imagine how, I mean, maybe we'll finish early.
It's such a time saver.
Right?
It really is. I remember we were doing that. What's that?
I should have thought of this sooner, yeah.
Yeah. Because I was thinking, when we were doing the mock presentations, it was like a guarantee it was going to
Take forever.
Yeah, a little bit.
And someone would have a problem and then you'd be like, what do I do?
Yeah, exactly.
I don't know who's first. Who's first? Seth's first.
Oh, that's right.
Yes.